Good morning, everyone. Welcome to Informatics Institute Bioinformatics PowerTalks seminar series. Today, I'm so pleased to invite Dr. Yan Guo here. Dr. Guo is a professor at the Department of Public Health and Sciences, University of Miami. He's also serving as a director of Biostatistics and the Bioinformatics shared resources, right, for the Sylvester Comprehensive Cancer Center. So which is one of the designated comprehensive cancer center in Florida. So Dr. Guo has a substantial experience with NIH-funded projects and has served as a bioinformatician on more than 40, probably the number can be updated, NIH-funded grants, including SBAR in lung, breast, and GI cancers. Before joining the University of Miami, Dr. Guo was an associate professor at the Department of Internal Medicine, Division of Molecular Medicine, University of New Mexico. He also served as a director of Biomedics Shared Resources for the New Mexico Comprehensive Cancer Center. Can I say that you funded the Biomedics program there? I know, right? Yeah. So, yeah, early in his career, Dr. Guo worked as a bioinformatician at the Vanderbilt University Ingrave Cancer Center. From 2011 to 2017, Dr. Guo worked as an assistant professor at Vanderbilt University, Department of Cancer Biology. Dr. Guo's research has been focused on the development of bioinformatics methodology and the analysis for genomic studies, and has published more than 200 manuscripts in related fields. So his work has great impact in the field. Today, his talk is Genomics Frontiers, Navigating Cutting-Edge Data Analysis in the Era of Precision Medicine. Please join me in welcoming Dr. Guo. So, yeah, it's your time.
Thank you very much for the introduction. I will share the screen. I will explain my talk. So can you see my screen, the full slides?
Yes, I can see.
Okay, all right, thank you very much. So this is the title of my talk. Thank you for the opportunity to speak here. And here's my email. So if you want to contact me regarding anything that's unclear today or any other reason, please contact me. First, I want to just talk a little bit about myself. So I've been always in the field of computer science. My education, at least, is. So I had a bachelor, master, and a PhD all in computer science. So I look at the problem a lot of times, not from a biological point of view. A lot of times I'm trying to solve it as a computer science problem. So that's just me. And I have always been worked in the shared resource. So basically, shared resource is a service. So the service, our job is to help other people to conduct their research by providing analysis. So this type of position, there's a certain advantage and a disadvantage for this job. The advantage is, I was able to, I was exposed to a lot of new technology constantly. So people keep trying new technology. So I keep getting exposed to it. So I get to know them earlier. I can get to explore them, developing methods, that kind of stuff. And the disadvantage is there's no focus. Actually, if you look at my 200 publication, whatever you do, there's not really a focus on anything. I go with the data. So whatever data I have available to me, and then I just try to think of what kind of problem can solve, what kind of computer science algorithm can be applied to it. So that's my usual thinking. So I was thinking, what should I talk today? Then I decided finally, I'm going to talk about 3 projects that's ongoing or near finishing state in my current research work. So we're going to talk about 3 different projects here. And they're all sort of deep learning related projects, deep learning application, except the third one. You'll see. So let's start the first project. The first project is about the convolution of genomic material origin. So regarding this, so look at this first paper here. This is the paper, original paper about cytoscape. No, not cytoscape, cybersort. Cybersort is a tool that can deconvolute RNA. Then we know what kind of cell type the RNA is from. So based on the gene expression data alone. So this is a deconvolution of RNA to trace back to the cell type. And then this one is another method for deconvolution of cell-free DNA from methylation data, from DNA methylation data. So they have DNA methylation, they can deconvolute. They say, okay, this data is from 40% of lung, 20% of kidney, that kind of thing. They can trace the origin of the DNA. Cell-free DNA in this case. And there's another one is for actually cell-free RNA. So they can deconvolute the origin, the tissue origin of cell-free RNA. So based on those, right? And then, so how did those work? How did most of this, all three of them have one thing in common? So they always use a method called, or similar method called non-negative least square type of regression. So what they need to use this method is they need to build a reference panel. So let's, for example, we have a panel from tissue 1 to tissue 10. We have 10 tissues, and then we have 50 genes that can separate those tissues. And then we build this panel. And then the next time we have a new sample, we sequence it, we have their gene expression, we know those 50 genes gene expression, we fit this method, we get contribution from those 10 tissue. So this is how this program works. It's based on a reference panel. And then similarly, if you heard of a mutation signature, this is also based on the similar idea. And also, if you have heard the PAM50 method from the to use for detecting breast cancer subtype. This is also a similar approach. It's always based on a panel. So what is the disadvantage for building a panel? So the disadvantage for building a panel is, for example, if we have many samples from tissue 1, from lung, for example, then in order to get one number here, you need to take either the average or the median. So by taking the average or the median, we actually lose information. We lose the variance information in that case. So we don't capture the variance in the reference. So this is my thinking here. So how can we improve this? So this is my original thinking. So I started working with one type of RNA called a cell-free circulating RNA, cell-free circulating tumor RNA, actually. So what is the cell? We call this CTC. This is cancer cell to, what is cell tumor cell, right? CTC, circulating tumor cell RNA. So what is the circulating tumor cell RNA? It's similar to cell-free RNA, but it's different. So cell-free RNA is from tissue only, but this CTC is actually from tumor. So if we have an original tumor, for example, we have a skin tumor right here, and then the tumor RNA get detached into a bloodstream and then travel into another organ and then become a metastasis tumor. So this is how tumors spread. So we can capture, there's a protocol in the biolab, we can capture such a tumor cell first, and then we can extract RNA from the tumor cell. So I was thinking, can we really, capture those and then extract RNA and then deconvolute it so we know where is from, or we can detect from here. So there's a term called organotropism. So this is this term means clinical observations suggest that the mouse cancers metastasize to specific organ. So once the cancer is metastasized, so it's starting to sort our RNA cells from this side too. So can we tell if there's some RNA from this side, so we can detect this earlier metastasis. So I was hoping for that. So, and how can we do it better than a panel? So this is the motivation. So what we did is we used a neural network approach. So this is the neural network approach right here. This is a traditional non-negative least square approach based on a panel. And then in the neural network approach, we actually use each individual sample in building this neural network model. In this case, we actually input each individual sample into the model. We don't have to take the average. So in that sense, we can capture the variance in the training data. So I was hoping this would generate a better result. So we started from those about 16 different tissue type right here. And then we used the GTAX data to identify tissue-specific genes, and we validate them in the normal tissues of TCGA data. So we identify those tissue-specific genes, and then we use them to build this model. So this model actually is just standard neural network. We didn't use any fancy model in this case. So we conducted several independent validation. The first independent validation is from GTAX. You know, GTAX is from donors, right? A donor died in an accident, and their tissue get a harvest for sequencing of RNA and DNA. And then usually one donor can donate maybe 30 to 40 different tissue types. So in GTAX, there may be around 400 to 500 donors, and then there are thousands of different tissue types. So what we did is we did a semi-in silico data set. What I mean by semi-in silico is from one patient, because the patient has multiple tissue sequenced. So we just took random tissues from that patient. We made combined tissue type RNA-seq data. It's like a fake RNA-seq data, but it's not completely fake, not completely simulated, but it's combined from multiple tissue at a different percentage. So we know the percentage contribution from each tissue type, and then we can predict the tissue type based on our algorithm, right, our model, and then we look at the difference between the predict and the real one. We know the real answer in this case, and the result was quite decent here. And then the second one is actually I went to the tissue repository back in New Mexico. I asked for normal tissue instead of cancer tissue. So actually, we obtained 6 different tissue types, and then we did a similar thing. We mixed the tissue type into different combination. We sequenced it, and then we also could conduct a semi-simulated data set. And then the third one is the most interesting one. We had one patient who is a skin cancer patient and had a metastasized tumor in brain and lung. And we had longitudinal data on this patient at month 0, month 3, and month 9. So we had different time points. 0.6 and not 3, and then we look at the composition of the percentage from brain and lung. We can see initially we detect very little brain and lung material, and as time going, we detecting more brain and lung as the tumor metastasizes to those sites. So this is an indication of a clinical application of this algorithm. So this paper has 2 limitations. This study has 2 limitations. So one limitation is actually the one patient is quite small, but in order to get more patients, it's quite difficult in this sense, but we require probably other funding and to get more patients in this case. So another thing also bothers me a little bit is, are we really detecting tumor RNA? So one of the biology involved here is, for example, if we have an initial skin tumor right here, and this is actually skin tumor cell, and then travel to, for example, here's the brain, and then metastasized here, and this is supposedly biologically still a skin tumor. would that just become a brain tumor in that sense? So I actually asked a few oncologists about this question, and they told me that it's possible, this is a brain, this may cause the brain to shed more brain tumor, so there's a possibility here, but no one is 100% sure in this case. Right, so there's still some question related to this. So based on what we found, but definitely the conclusion is neural network is better than the traditional regression method because it accounts for each data points during the training. So we don't have to based on a panel, right? Depending on the panel. So there's a potential clinical application for this method. So we can detect early metastasis tumor. If this can be real, it could be very, very good for monitoring patient. So this manuscript is currently in revision. So let's go on to the second study. The second study is about gene expression prediction. So gene expression prediction sounds very trivial, right? Why do we want to predict gene expression? So sometimes I ask this question as well, but if we look at the literature, so this paper is from 2016 or 17, published in Cell. So they published a method called L1000 method. So what is the L1000 method? So they're claiming basically they can predict almost the entire transcriptome, actually not, maybe half the transcriptome from 1000 landmark genes. So this is what they did back then. And then they built something called a gene connectivity. And this is a large consortium. It's still available right now. So the method actually just used a traditional linear regression for prediction based on those 1000 genes. So how they trained the model is quite interesting, also maybe questionable. So what they did is they collect this many, 12,000 samples from GEO. So basically they collect samples from all tissue types, all conditions. They didn't care. They just collect all of them and then build this model. And then they build this, and then they use this. This is actually the recall. So they build a random background. And then they look at the correlation of those genes, and if the predicted gene's background is greater than 90%, 95% of the random background, they say, okay, this is a good, significant. So this is how they determine it. So what they didn't do it is, I thought about this. There could be a potential limitation here, is they don't tell us the actual correlation. They just tell us this is better than 95% of the background. The background could be very bad. The background could be, the correlation could be 0.3. Highest correlation could be 0.3, and then if your correlation is like 0.29, that's not very good. So I thought we should just also based on the real correlation and in addition to this recall definition. Also, another assumption they made is the linear regression was used, but gene expression may not always be linear. So there are other mechanisms in gene expression regulation. Could be a threshold response. For example, microRNA, when microRNA regulate gene expression, is usually based on a threshold. So it's not that linear. Also, a situation called such as feedback loops, they will cause an oscillation in gene expression regulations. And there's another article, I think in 2001, they actually predict the gene expression just from sequence. So they can predict gene expression from one piece sequence to another piece of sequence. So this is based on a convolution neural network. And then, so I thought about those two, and then I thought, how can we do it better? So here is what we did. So initially, another limitation is, I thought potential limitation in this original paper is when they combine all those samples together, so their normal sample, their disease sample, their tumor sample, so the correlation must be different between different conditions, right? So we did a brief analysis here. So we took a TCGA normal, we took a TCGA tumor. In this case, we just took the breast cancer tumor. And then we took CCLD breast cancer. CCLD is a cell line database. And then we took a GTAX data as well, somewhere right here, t-tag is normal. Then we look at their correlation of correlation. So what do I mean by correlation of correlation? So this basically means we have a pairwise correlation within one data set, right? Within one data set. If we have a gene A versus gene B has very high correlation in g-tags, do we observe similar high correlation in another data set of the same pair, gene A, gene B? So, and then we look at the pairwise, and we look at those two pairs. So this is the correlation of correlation. So obviously, when you look at the normal and pair with normal, we have a pretty good result. And then when we pair tumor with normal, we have a less good result, right? This is it's very intuitive, right? So, and then we look at breast cancer. It turned out, for example, CPTAG is another large consortium. They have breast cancer, and then they have a very, very strong correlation. So in tumor, so how can we leverage those information? And then we also look at other consistency here. I'm not going to go into too much detail here. Let's go on. So this is our study design. We decided to use a graph neural network approach, and then instead of a traditional linear regression. So those are the data set we have. So in the training, we had t-tags normal and the TCGA tumor. And in the validation, we used TCGA normal, CCLE cell line, and then CPTAG tumor. So this is our pairing scheme. And then for the models, we tried traditional linear regression. We tried multi-layer perceptron. This is just traditional deep learning. We tried autoencoder. We tried two different graph type of neural network, convolution network and the principal node aggregation. So this is the rough depiction of the network, graph neural network. So features. So traditionally, in linear regression, in the original paper, they only use the gene expression. So can we use more? So we use the spatial information, and the regulation information. So here we actually used 2D genomic distance and a 3D genomic distance from high-C data here. And then we used the pairwise correlation matrix. Also, we used other regulation I didn't put here, including like a pathway network, that kind of information. So we evaluated them by correlation, also by recall, which is the original paper used. Also, we used the mean square error. We used the 3 different matrices to measure it. So this is the result. And then long story short, right? So the neural network-based method usually can perform better than linear regression in this sense. So to make it fair comparison, we used exactly the same genes in the original paper for this prediction. So they selected like 955 genes in the original paper. We used exactly the same 955 genes for our prediction. So our method can predict with more accuracy, not accuracy, recall in this case. This is the recall, and this is the correlation, and with recall, this is another measurement of percentile rank, and this is the mean square error. So it shows that actually the neural network can perform better than linear regression. And that's because neural network model can account for additional information. So how does those additional features help? So we did some additional analysis. So right here, if we look at a simple regression model here, so this regression model is between the 2D correlation and the 3D distance, 3D distance from high-C data. So if we look at this correlation, and we use a different regression, nonlinear, this is a linear, and then we use the linear regression. And then we found, yes, there's correlation between gene-to-gene correlation and there's 3D distance. And their distance and 3D, this correlation may not be linear. So although linear is significant, and the other representation of these regressions are also significant, or more significant in this case. Then if you look at this is arc plot, we look at the top 500 representation from linear regression, not from the gene-to-gene correlation of the 3D distance, they're quite different. Now, this is a little bit hard to tell. So we draw another graph here. They're the same genomic location on the X and the Y. And then the one color is the gene-to-gene correlation. The other color is the 3D distance. And then we look at those hotspots. They actually are not together. So in the end, if we look at this way, if you look at top, how many pairs, how many genes, and then we look at this number of how many genes, and their highest overlap is only 3.5 to 3.5%. So what this means is really the 2D information and the 3D information, they're offering different information. So we're capturing different information by including the 3D data. So this is a general result. So this is by different combination of what we were inputting into the data, we're getting different results. So different combination of those data can generate different results, and then sometimes better, sometimes a little bit worse. And then those are the two graphical representation, GCN and the PNA. And the PNA performed a little bit better by GCN. We're still refining these results. So the conclusion is the graph neural network has better performed because it can incorporate additional graph represent features. And then 3D structural genomic capture additional layer information not possible by 3D, not captured by 2D. And the regulation network also actually we input pathway information are also useful features for building this gene-to-gene connectivity network. So this manuscript we're preparing, we're near the end of this manuscript. If you have any suggestion, please let us know. So additionally, we also tried other regulations such as like transcription factor regulation, RNA binding protein activity regulation, but those had less effect, so we did not include in this study. All right, so on to the third study. The third study, the last study, is about HLA. So for those of you who study or know HLA, HLA is also called MHC sometimes, and it's a very important set of genes that regulate the immune system. So if you look at HLA, they are on chromosome 6. They're actually located right here on the P arm of chromosome 6. And they're a set of genes starting from A, C, B, and a set. So there's this figure on at least 6 genes. There's actually more than 10 of them. And then if you look at the HLA genes, this is usually the annotation of an HLA gene. So starting with HLA and A denoting the HLA gene, and there are 4 different level or class of HLA genes. So the first class or level denotes the allele group. So each person have 2 alleles of HLA, and then second one denote specific protein, and the third one denote synonymous substitution in the protein coding region, and then the last field denotes non-coding region, changes in non-coding region. So most people only care up to the top 2 level. They don't care about those. So if we look at all of the HLA genes currently available in the database, for example, if we plot HLA A genes out, the structure looks like this, and then there's actually tens of thousands of sequences of HLA genes spread out on all 4 levels. So how can we detect HLA gene? So we want to detect HLA gene from RNA-seq data. So there has been maybe 1 or 2 papers talking about detecting our HLA from RNA-seq data. And then we thought we could do a little bit better. So we actually determined an algorithm for doing this. Initially, I was using a very naive method. So basically, I built a HLA reference, including all HLA sequence there, and then I just do the alignment, and then I make sure I did not allow any mismatch, and then I just added them together and see which one get represented the most. This turned out to be a very wrong approach. We cannot do that. So the correct approach, what we came up in the end is like this. So we have reads aligned to different class, different set. We call them 1, 2nd, 3rd, 4th, OSTF in this case, and then for this set, for example, for this level, so those are the set operation. This is not a minus. This is the set operation. So F minus here, F2, that means we're using a unique, we're getting all the unique reads from a non-F minus F2, not in F2, and then we use F2 minus that. In this case, this set operation will return all the unique read mapped to F2, and then in this case, then we can get all the unique read in F2, and then we can push back to infer the original this HLA allele, and then so on. This algorithm works in such a way. So how do we call the HLA, the genotype? So for those of you who understand, this is very similar to how we call a SNP or a mutation in some cases, from sequencing data. So it's a Bayesian approach. This is the formula right here. Let's just give a brief example. So if we have a 33 reads that support HLA-01, and then another 6 reads support HLA-15, and then because it's a Bayesian approach, we have a prior probability. If we don't know the prior probability, then we can just assign the 3 genotype to be 33% each, and then we can set an error rate to be 2%, and then we plug in all those numbers in the equation, and then the genotype for this HLA-A to be homozygous 0101 is 94.8%. So based on this rate, where it's intuitive. So this is the standard approach. They use that in the SNP analysis as well. So based on that, we also construct a distance, or you can call it a distance or a similarity matrix within one class of HLA, not within one class, with one type of HLA. For example, HLA-A would have a matrix, and this matrix is built based on this kind of algorithm. So we have a pairwise matrix, distance matrix between any 2 HLA sequence. But sometimes the HLA, this level, doesn't have a specific sequence because those are represented by the lower class sequence. So this is actually not available. So we can simulate it, push it back up. So it's like a backward tracing algorithm in this case. So once we have that, we can compare HLA difference between 2 groups of samples, and then we adopt 2 different algorithms. The first one is Adonis. This is already established algorithm, and then it's basically compared to matrix, distance matrix in this case. And then the other one is our algorithm. We build a permutation algorithm. We can build a permutation matrix many times and look at how many times it's greater than the real distance in that case. So this is the algorithm right here. So another way we can do it, we can actually look at the HLA binding sequence. So because we know the exact sequence of HLA, and then we can know their binding motif from a database, and then we can check against other specific binding protein or sequence. I'll give one example how this is done. So right here, data set 1. So this is a real example. We have 3 different data sets for this analysis. So in data set 1, we have this cell line A549, which is a lung cancer cell line. We have none of them. So they're run in triplicate. And then the first set was treated with IFN, which is interferon. And then the second one is treated with flu virus, just regular flu virus. And then the third set is just marker or control. And then we perform the RNA-seq. And then we put them together, three of them together, we performed isoseq on those data, those pools. And then we applied our algorithm on it, try to detect RNA-seq, try to detect HLA, and then those are here are the results for the RNA-seq. So I only list the first 3 genes of HLA because there's no room to list everything. And then overall, those 9 samples has a consistency of 93, almost 94%. They're from the same cell line. So because they're the same cell line, their HLA should be the same. So this is a good validation. And then if we compare our result with isoseq, so and this consistency is 100% consistent with isoseq data. So the first data set basically serve as a good quality control for the methodology. Just look at the second data. Oh, we also performed this analysis is called alpha diversity analysis. So another method we can apply to this, our result is alpha diversity, and we can compare those 3 conditions, and then we can identify a significant difference. And those significant difference actually does make some kind of biological sense to me. And then between the 2 treatments, they don't have a difference, but between the treatment and the control, we do see a difference. So I still need to really talk to the people who generate the data, talk to my collaborator, see if this makes complete sense. So data set 2 is a data set about multiple sclerosis. This is a disease also involved, it's an immune deficiency disease. And then this data set has 233 patients, and then has 69 control, and then have 2 different multiple sclerosis subtype, 58 and 106. And then we used our method to call the HLA, and then we used a method to compare the difference, and then we identified those differences. And then those are the differences we identified. And between the control and the diseased group. And then we also identified some other things between race and gender. And those things we were still examining it to see if they make any sense or not. But those data are potentially very encouraging when I talk to the people who provide me the data. And then when we compare our result to the Adonis, in general, this is the p-value, and then they're very consistent. And occasionally, one method is more sensitive than another method. So for example, this case, our method is more sensitive than this method. And then if you take this comparison out, we look at this, and then this is the blue part, this matrix right here is the control group, and this part, the green part, this green matrix is the disease group, and they do look differently. And this is probably not the best way to represent this. I'm still trying to think of the best way to present this result. Still working on this part. And then if you look at the alpha diversity, and we can see the difference between different groups in terms of alpha diversity. And then this is also another encouraging result. We're probably going to include this in the final. Now, the third data set. The third data set is probably the most interesting data set. So in this data set, we have 39 patients right here. And then this data set is unique because it has 26 American Indian patients right here. So American Indian patients is very rare. We had to get a special ARB, special permission to collect data from them. And then we had 8 Hispanic, 3 non-Hispanic white, and 3 unidentified. So all of those, we have those patients are COVID-19 patients, and then they had a response to COVID-19, and we classify them as a severe and non-severe based on certain clinical information. And then we didn't define this. The doctor defined this. They had in the medical chart, they had this definition as severe and non-severe. So we have this severe and non-severe phenotype. And then we had the information for those patients from day 0, day 1, 2, 3, 6, 9, and 14. And then we collect blood data from those days on all of those patients. And then we perform RNA-seq. So we have a longitudinal RNA-seq data on those patients. And then not every patient has all like those 7-day data, but on average, each patient has maybe 4 to 5 days of data. And then first thing we did is because those are from the same patient, right? We have multiple data points from same patient. We look at how consistent is the HLA call. And then the overall consistency is 0.97%. So this is a very good accuracy about our algorithm. And the next time we were looking at whether severity and non-severity or race have a difference, between in terms of those HLA genes, and we did determine many difference for different HLA genes. So this can potentially explain why those patients had more severe response to the COVID-19 virus than other people. And we look at alpha diversity as well. So the alpha diversity is non-severe people usually have more diverse of HLA genotype. So this result is actually consistent with the data set too when we look at the multiple sclerosis. So the control had more diversity than the diseased. So non-severe response had more diverse HLA. So maybe this is an indication that more diverse diversity in your HLA is better for the immune system. That could be an indication. We are not making the conclusion yet. I still need to do maybe a little bit more or talk to more immune experts to make a conclusion here. And then the more interesting thing is we conducted a motif analysis. Right now we know exactly which patients had what HLA sequence. Also, we know which COVID-19 virus the patient had because we did tests. We know roughly the strength of the COVID-19 virus. So we can see if there's more likely binding for what patients. So the result is here. So these are for the B and C genes right here. So for the B gene at least, non-severe people had more binding chance than the severe patients. So what this means is when the HLA binds something, it attack it. It treat it as an invasion of a foreign, material virus. That means the HLA identified it. So what we did here is we matched HLA against the spike protein of the COVID-19 virus, right? The spike protein are used by the COVID to invade the cell. That's why majority of the COVID-19 vaccines are designed based on the spike protein, try to identify the spike protein. So that means the people with this type of HLA sequence are more likely to identify the spike protein of the COVID-19 virus. So this actually, in a way, explains the severity and non-severity of the patients. So this is quite interesting. And then also regarding race, because the sample size, we only have 2 and 3 here, so I wouldn't read too much into this race. But it's probably reasonable to assume that each race have a more likely different HLA haplotype. So that will be a reasonable assumption. I need to do more research on this one before I make a conclusion. So what is the next step for our study? So this study is ongoing. We're near the end, but there's still more we can do, we want to do. One of them is doing a contrastive learning neural network comparison. So in addition to the Adonis and our permutation method, there's another deep learning method called contrastive learning, and then we can compare two different matrix, do a self-supervised training. So we're currently implementing this. Another one is we can utilize this. This is another method by Shirley Liu called Trust4. This one can identify the immune repertoire sequence from a single cell RNA-seq data. Basically, they can identify the CDR1, 2, and 3. Those are antigens, and then in a patient. So we can actually modify this to make it work on regular RNA-seq data, and then we can identify, for example, for the multiple sclerosis patient, we can identify their sequence here. Then we can look at their like HLA binding potential with those sequence, their specific sequence, and then we can correlate it to their treatment response and that kind of stuff. And another thing we're trying to do is right now we're looking at each HLA gene individually. So, but HLA is a haplotype. By haplotype, I mean certain genes are acting together, form a haplotype together. So we're trying to extend our method on a haplotype level in this case. And I think this is all the method examples I want to give. And then here's my lab member, Li Minjiang, is a research assistant professor in my lab. Yongli is a staff programmer, and Feng Yao is also a staff scientist. And then we have a collaborator, and all of those projects actually were initiated in New Mexico. And Dario Marchetti is an expert in the CDC RNA field. SkyNet actually helped with collecting the normal tissue RNA. DJ Perkins is the PI actually of the COVID-19 study. He was able to collect the American Indian data. Stephen Baker is the one that produced the isoseq data. And then Chase Spurlock is my previous colleague from Vanderbilt. He now owns a company, and he provided me the multiple sclerosis data. So thanks to those people, so those studies are available. And then hopefully it's informative, and also I'm hoping if you can give me any suggestion on how to improve those studies, I'll be very grateful. Thank you very much.
Great talk. Thank you, Yan. Especially this work has not been published, and one is in revision and the other is yet published. So I really appreciate it. And I hope in the following discussion that you can hear some thoughts from the audience. So I saw Steve is here. Welcome. So you can feel free to unmute yourself. Yeah, he was asking what is the PNA?
PNA is called the full name of PNA. It's called principal node aggregation. It's a type of graph neural network.
I have a question about the first part of the talk. So you're using the neural network to do the tissue deconvolution.
Yes.
So you use the GTEST data to create a simulated synthetic mixture. So I wonder why you use only 6 tissues to create the synthetic data, because there's almost 50 tissues in GTEST. Is that because there's a limitation on how many tissues you can handle?
Well, the reason, so we did this already, so think about it this way. This is the GTEST one patient produced multiple organ tissues, right? So in a way, this is a more realistic simulation in the real application, because in the real application, a patient's CDC data is from multiple organs from the same patient. So we're simulating this. This is a little bit less because when we collect the tissue, those tissue are not from the same person, right? They're from different person. In this, it adds more noise than this data set.
You can identify which tissue from the same donor.
Yes. Well, I got this from the New Mexico tissue our own repository in the Cancer Center. So we don't have, we have some limitation of where we can get it. So we only got 6 tissue. We didn't verify whether they're from the same person, but most likely they're not from the same person. And then this is a more noisy simulation or semi-simulation of the data in that case. And then I didn't show the final result figure. And this is more consistent than this. So this is still good, but it will be less good than this, because this is more noise in a sense.
And for these CTC cells, how do you determine they are CTC cells?
Here's a completely wide lab protocol for this.
So there's markers or something that they collect.
They have an assay for it. They have a special kit to extract it first. There's the person, the Dario Macchetti, my collaborator, also had a patent, like a register. He's trying to register this. But I always, be more spectacle. Like I said, this really detecting tumors are, I always, a little bit, suspicious of this, but at least I can conclude, this neural network approach is a little bit better than the traditional approach. That I can pretty safely say. But whether this patient data is just a random chance we observe it or is real, I cannot say. We need to do maybe a lot more to know for sure.
Sorry, just a follow-up question. So for your test, so you list the brain, lung, and the skin here. So how many tissue did you test there? Is there a limit on that?
For this?
Yeah.
For the patient? Oh, the patient just one sample. This is a CDC, it's a cell-free.
Right. But how many tissues?
Oh, we run 16 tissues. 16 tissues. Well, model is for 16 tissues. The 16 tissues right here. So initial selection the tissue is pretty tricky because if you look at the GTAC, there are actually more than 40 different tissue types. And then the first filter is what tissue type cause tumor. When we're just looking at that, then we filter out all the tissue type that do not cause tumor. And there are tissue types that do not have tissue-specific genes. There are certain tissue that does that. So those tissue that doesn't make sense to put in here, or they just really make noisy things once we add them. So they just exist in everywhere. So it's making it really harder to conduct this. All right.
Any other questions from the audience?
Hi, Dr. Guo. Very nice talk and great studies. I have 2 questions. First one is for the deconvolution project. So some study, I believe the one you show in the data communication, they argue that the data linearity is kind of important for to get a good deconvolution result. For example, I believe in CyberSort, they suggest to use the RPKM instead of the log2 transformed RPKM. So my question is, do you think this is, this kind of sort of linear scale data is, the data linearity is sort of preserved in your neural network model, or you think this is not a concern?
The reason, I talk about linearity a few times in the next 2 in the example 2 also. So I think one advantage or potential advantage, I can't say for sure, all right, for the neural network model is it doesn't assume linearity. So if there's a linearity in the data is captured. If there's not linearity or other type of association is also captured. It's captured through the structure of the model, all those edges, between the nodes, that kind of stuff. So it's captured very intrinsically within the model. So that is, that's my belief, at least. I think that way, is a little bit better. That's why it's not a concern. I think we don't have to assume linearity in this case.
Okay.
Well, I mean, but the data could be, if you apply a linear model, it could be entirely correct. If the data is really linearly associated, is entirely possible, right? But I don't know, if this deconvolution, I don't know if we should assume a linear, because it's possible. I can't say for sure 100%, but I assume the graph, the neural network it will be linearity agnostic.
Okay. So for your second project, gene prediction, maybe I missed that, but what is the tolerance of, I mean, for the L1000, right? They start with 1000 genes.
Right.
And your model, is your model also starting from the similar number of the genes?
They started with 955 genes. We started with the exactly same 155 genes.
If you compare this model, there was some sort of dream challenge project where one of the things I remember is about the data imputation. But I believe that one, probably the gene expression prediction is only for slight, smaller portion of the genes. Have you compared the performance of this and yours to those imputation-based model?
Yeah, well, we didn't compare it, but what I can tell you is, the only reason we use this 955 genes is because we want to compare it fairly with the original paper. That's why if we were to select our own genes, we will select a different set of genes based on a different set of criteria, or like a dimension reduction technique or other technique. And we can predict more. We can already predict more than this number right here. We can substantially decrease this and increase this number. But the reason, I guess the point we're trying to make from this study is, by first, neural network can be better, and by adding other graphical features, it's also useful. So those are the two major points. So we're not trying to, to make a model for people to use in a sense. But in a sense, if we do want to just build a model so people can use freely, then we probably wouldn't use this original 955 genes. But you make a good point. We probably should make the best model available, just to say this is the model, the best model, if we were to build it.
Yeah, I think that would be another probably interesting project. I mean, because I guess the original reason they come out with this L1000 is they want to, save the budget.
Save the budget. Yeah.
Save the budget for this large scale data generation. And if you can have, based on your method, have another set of genes, this might be have better use.
You make a good point. The reason I didn't do it, because I always had the impression that gene expression prediction or imputation is not that useful. Because most people do RNA-seq, they get everything. It's cheap enough. People don't need to. This L1000, probably is outdated. I doubt they still use it. So I'm trying to make a point from the methodology rather than from an application point.
Yeah, but for other omics data type, like proteomics, for example, for the phospho, I guess they also have some another phospho platform that might be even more useful.
You're right. Actually, we're doing another model for methylation. It's more complicated than the gene, because the number of CpG is substantially larger than the number of genes, and then make this traditional approach, even those graph approach, are impossible. We had to come up with other approach for that.
Yeah. Thank you.
Great. I saw Qian is here also, so good to see you, friends. So I think we are running out of time, and this is great talk, and especially this has yet to be published. And thanks for the great questions from Steven, Qian, and other folks. So I think we can stop here, and hopefully sometime we can meet in person. And thank you, guys, and take care.
Right. Thank you.
Thank you.
Bye bye.